I will give you a lot of dots and you can connect the dots. So my background started in wet lab biology, pure hardcore microbiology, molecular biology. So until I finished my PhD, I don't know anything on bioinformatics. So I finished my PhD in University of Madras in 1996, 91-96 batch. Conventional cloning, purification of proteins, that's a big thing in 1996. Even one ELISA was a very big thing in 96, even at Chennai Madras University, where the quality is very high compared, there is only two places in those days, Madurai Kamaraj University and EC Tech campus in Chennai. So did the best science, but still figure it out that it's a very challenging field. Realized that in the third year of my PhD that I need to do something. So I started to do my GNET during my last phase of my PhD and moved towards computer science. So I finished my PhD and I got two postdocs purely on vet biology. One in Australia, one in US. And that is a normal culture in those days in AC Tech campus. You will get a postdoc, no matter, because the quality of lab is like that. So it's not on me, it is the professor's background that helped us. And if you know some of the people's name like Professor Magadevan, Professor Dharmalingam, these are all the stalwarts in that time. So we were lucky enough to go through their training and we got the placements. The challenge for me was that I realized that, okay, this is not challenging to me because that time it's a one gene, one protein hypothesis. Central dogma changed now. If you know the real biology now, the central dogma changed in the last probably 20 years when we understand that there are multiple splicing because of transcripts and multiple isoform proteins. But in our PhD days, it's a one gene, one protein. So even when you do one clone and then one purify one protein, it's a big thesis. So I realized that I have to move to the data science, bioinformatics. And in 1996, I applied for the Wellcome Trust Fellowship. So this is where I'm giving some dots. You have to think out of box. Don't go with the traditional methodology, what people are doing. You are coming from a very strong B.Tech biotechnology, which is a mix of jack of all, what we call, because I come from a BSc MSc background those days. You people are already in the technology background, so I think I was talking to Dr. Manimaran. You have a lot of flavor on physics, chemistry, maths inbuilt into your career. I don't know how you are leveraging it, but this is where you need to think out of box. So being from a science, I went to computer science and I will explain how far I went. So being a B.Tech biotech student, don't go with a traditional what the industry as a biotech is looking at. Think out of box and I will give you some pointers where the industry is currently in demand. So it's always what we call the demand supply. If you go and then apply us with a biotech, B.Tech or M.Tech or even a PhD from Indian Institute of Science, where we have seen a lot of CVs doesn't qualify to the best of the industry. The reason is simple. They look at only two things. What is your skill set? What is your problem solving ability? What you can do in the current market. So if you do your current traditional biochemistry, or I'm not blaming the domain, but you have to use your domain, but you have to apply your technology there. So a hybrid is must. Minimum two field you have to know bring together on table and solve a problem, not with one field. So if you are good in biochemistry, think about something in data analytics. If you are good in, chemical engineering, then something where you can integrate a robotics with an automation for the upstream and downstream. I'm throwing some lights what you can think about. So coming back to where the AI is emerging, I will walk through in 5 minutes about the history of how the data changed. So in 1996, when I started my bioinformatics as a postdoc through the Wellcome Trust International Fellowship, and I was offered at MRC Medical Research Council at Cape Town, and this is one of the 5 positions in the world. So I was the earliest bioinformatician in the world. So when I went there, I don't have any idea that the field of bioinformatics will change this much in the last 25 years that I have traveled. I didn't understand that I am part of a game where a lot of transitions are happening. All I did was, I had a passion, and I know that this is where I need to move from my traditional biology and then do something on top of my biology and do something extra. This is how I started. So in 1996, my first work was on Mycobacterium tuberculosis. And TB is a predominant African communal disease. And Wellcome Trust is throwing millions into the African continent, and my job was first to annotate and curate the Mycobacterium tuberculosis and Mycobacterium leprae, which we were doing a joint project at Sanger Institute at Cambridge. So that gave me a first opportunity to understand what is bioinformatics, how people are working, what is the processes, what is the SOPs, what is the methodology. All those things I learned in 1 year. Then 98 I moved to Human Genome Project, part of the international consortia at South Africa, and that is my best breakthrough that I could expect. So went to build the first human reference genome was built by our lab, and I am one of the author in 1998 itself on the human genome. Which is called a stack database, which is purely an algorithmic approach for assembling the transcripts of the human in those days. A lot of gaps were there, but that's the first attempt in the world. Having published that in IEEE and all, and I will tell one moment that changed my life. Okay, this is all some sparks. So when I was working in the SANBI, the South African National Bioinformatics Institute, I had a colleague called Peter Van Hoosten. Still I remember him. He's a Dutch guy. So I was struggling to code at that level, honestly, because being from a hardcore biology, it is not easy to scale up in 1 year and then do a C programming at assembling a human genome. I was really struggling. I was able to do patches, but I was struggling, and I wrote a code and that crashed the server. And the server is at the NSGR at US, where we had to do a VPN connection. So he told in the group meeting that who taught you coding, who taught you computer science, in front of like 15 people. And that's the time I realized that, okay, I have to scale up my computer science skill set. So I started to move up from my conventional logical programming to the hardcore proper computer science guy. So again, I had to do a transformation. So I moved to Canada. I started to work with Chris Hogue. Again, you need to pick and choose your mentors at the correct time in the correct place. This is again very important. So I moved to Chris Hogue. If you Google Chris Hogue, you will understand he is the inventor of MMDB at NCBI. That talks everything. He is the PA of MMDB. He is NCBI senior scientist, and so there I learned computer science proper. And I talked to Chris, and I told that, Chris, I'm not able to go to the real algorithmic implementation, but I'm able to do analytical part. So again, he told that you have to go and then join a part-time BTech engineering at University of Toronto. We will sponsor you. So the point is, you have to work really hard. There is no doubt on that. 6 o'clock morning with a boot and a 20 centimeter snow, I used to go to the classes and then finish at 8:30 and come to the office. So 3 years, I finished my BTech computer engineering. That's it. That changed my whole perspective on getting the grip of the bioinformatics. Being a computational biologist, I moved to a bioinformatician. So there is a change. There is a definition between a computational biologist and a bioinformatician. Real differences: computational biologist will analyze the data with the domain knowledge. A hardcore bioinformatician will write algorithms, meaning we are part of algorithms like for you BLAST, Smith-Waterman algorithm or Needleman-Wunsch. How do you implement that with using the NCBI ASN structures, libraries? That's where the hardcore bioinformaticians will come into play. So Chris was being part of the NCBI. He gave all the SDKs and APIs from the NCBI. We developed the whole process at MTS Proteomics. So again, at that time, then there is a gap again. My statistical background. So they told that, okay, you have to do few modules in statistics, mathematics and all. So I went and then did my discrete mathematics, linear algebra, and then hardcore 2 modules of statistics, that's it. Then I moved to Genome Canada, where we have a great opportunity to look at a few, at least 20 genome in those days. Again, if you look at the history, why I'm talking is from 98, where even one Sanger sequencing of one clone itself is a big thing for analysis. From there, the field started to change drastically in 2000, at least, when the first assembly was released and when Sanger sequence was assembled to a decent level, and the first publication of the human genome happened in 2000-2001. From there, the field started to change drastically. One thing that you have to understand is when you go to the real tech space, you have to adapt very fast, otherwise you will be sidelined. You have to see in terms of tools, skill set. I started with Perl, then I moved to C, then I moved to C++, then I have to do Java. This is all tool set, but my biology is intact, and that's where I was able to go and then put the best pipelines. So then I went to the European Bioinformatics Institute, EBI, which is world's top 3 in the world, where still today NCBI, DDBJ at Japan, and EBI. This is the 3 places where every pipeline, data submission, standards, qualities, frameworks comes into place. So I was part of the human transcriptome project. So we wrote complex algorithms in Ensemble. I don't know how many people know Ensemble pipeline. That's a complexity on its own. This is still one of the most complex computational pipeline even in the world today, even today. So if you want to touch one Ensemble pipeline, you should have a very strong IT background. So I wrote the algorithm for the alternative transcripts. Again, if you look at it, is how do you transform yourself from computational biologist to a bioinformatician to a hardcore algorithmic person that can put modules into Ensemble framework. So the industry is now let me put a break there. Now let's come back to the topic of the day. AI is there in bioinformatics at least for the last 30 years. People are not aware of that. If you see the gene finding algorithm from Tim Hubbard group or any early algorithms that were still in place, nobody has changed even a single code on those written by Tim Hubbard, or there is one from Washington University. Still it is the golden rule. But only thing that has changed is those days the data is very minimal, IT infrastructure is very minimal, so we don't have an opportunity to work on complex data. Even RAM is a big thing. So even 126, 128 GB RAM is a very big thing in 2000, mid to 2000. Where the things started to change is when we move to the GPU era, when we move to the NVIDIA era, when we move to the so-called AI era, which I will come to AI era now, and I will connect how the omics platform is giving an opportunity for you for the next 20 years. So if you look at from pure bioinformatics perspective, there is a famous Moore's law in bioinformatics. Every NCBI slide you will see that. I don't know how many people have seen that. Moore's law is how long will it take to double your data. So bioinformatics started to challenge Moore's law probably in 2005 when Solexa started the deep sequencing. So later Solexa became Illumina, and we know Illumina is a market player. 85% of the market share is by Illumina. The way data coming from Illumina platform or from Nanopore, NanoString, PacBio, phenomenally high. So today in 2025, next 1 year, the expected data just from genome is 40 exobytes. I don't know how many people know exobytes. You put several tenfold after gigabytes, petabytes, exobyte is 40 exobyte. And there is a Nature paper again last year that the major challenge will be in the next 2 years is analyzing the human genome data from population and other perspective that is coming from all kind of omics platform. One of the fundamental challenges from an IT perspective is you have to understand the whole problem. How do you store the data? What is the data center required? What is the data size required? Which algorithm is good for your data size? And what is the questions that people will ask? I'm sure prompt engineering, you are aware of what is prompt engineering now in the era of ChatGPT, which I'll touch upon. But 2025, there are already 2 articles which talks about exabytes of data, and also there is a Nature paper again which says that the data that will come from human or from genome platform will be much larger than the social platform like Twitter or your Facebook and all. So you can imagine the scientific data that's going to come out in the next 2 years, and already IT challenges are being addressed by IT companies have started to come. So Wipro has a separate division now exclusively for genome in Bangalore. TCS has put a bioinformatics facility at ITPL in Bangalore. So already players at the top IT level have come to handle the data side problem. But one problem that they cannot handle without people like you is the domain problem. Yeah, they can work on putting a storage, they'll work on NFS structure, they'll have a NAS structure, they'll work on server structure, they'll work on GPU, but they will never be able to touch the bioinformatics and the AI algorithms. That's because it's a heavy domain problem. One challenge that will challenge you is, now there is a lot of hype. So let's now talk about AI. Okay. So bioinformatics is growing, data is growing. There is a lot of demand supply problem in bioinformatics, but everything changed when AI came. So people started to understand the power of AI. So as I said, Tim Herbert's neural network algorithm were there for last 30 years. Still, that is the best algorithm even today for all your gene finding. So if you go and look at the gene finding, and this is all written previously. Nobody has done anything. But what is changing today is the kind of data that we hold today. So AI, as you know, there is at least there are 2 flavors. One is your machine learning. The other is the deep learning. For a biotech people, I will put the definition like this. Machine learning is an algorithm where you apply and deploy already existing machine learning algorithms for the data that you have. And deep learning is one level above that, where you don't have to, so deep learning can work on unstructured data, put this way. Machine learning will work reasonably well on a structured data. What is unstructured data? Structured data for a biologist, example, if I have a protein which is highly curated like a Swiss prot standard, and I know my functional description of my single protein, that's a structured data. So if you give the structured data to the machine learning and then ask the model to learn and then predict, it's one way of doing a machine learning implementation. Where this will be applicable is, you have a new project coming in, and then you have a model built on top of the Swiss prot, and you can deploy the model and then ask the new model what is the output or what is the prediction. That's a straightforward machine learning. Deep learning, yes, if you see even in bacteria, 40% of the open reading frame doesn't have any functional annotation. These are all still mysterious, or people doesn't have any answer. So until you curate it, so the problem with the curation at the Swiss prot standard is, until you have a functional evidence and you have a gene knockout or you have a mice model that you are able to associate your functional characteristics of even a point mutation, you cannot curate a Swiss prot standard thing. This is the golden standard. This is why Swiss prot is even is ruling the whole bioinformatics field or even the pharma industry. They have a very tight, very strong standards for data quality, curation, and so I worked with them for part of the project. 45 top quality PhDs from the West were curating day in, day out for Swiss prot. And there is a big pipeline. But now, deep learning, what it is going to do is, you throw your data which is unannotated and which doesn't have any functional data, and this will learn on its own. But there will be one problem. Deep learning, you need a very big IT infrastructure, meaning your GPUs has to be like ChatGPT. The problem with the deep learning is you need what we call the tokenization of data has to be in few billions, not millions. So you have to throw a lot of billions of data tokens, and also in deep learning, you should throw as much as data which is unstructured. Then only deep learning will come into play. I will walk through a few examples. Okay. Now in the current scenario, let's come to the current scenario. If you look at any human genome from a diagnostic perspective, which is emerging very fast application-wise, because the whole genome is now costing us $700, $800, whole genome at a 30x coverage. And a whole exome is costing us Indian rupee-wise 10,000. This is a service provided cost. Your actual cost could be 6,000 to 6,500. But the challenge is how do you delineate and then how do you come to the actionable mutation points. So I'll walk through one case study. The AI is coming very hard into the whole holistic pharma industry, which could be genome, transcriptome, your proteome and metabolome holistically. But data that is coming exponentially very high is both your RNA and DNA data. In the DNA data, the fundamental question people will ask is, what is my mutation? So if you look at the ACMG classification, people clinically will classify your mutation into five classification categories. One is pathogenic, whether my mutation is pathogenic, likely pathogenic, benign, likely benign. The fifth category is unclassified. We don't know currently. So I'll tell one example, current scenario. Before 2 years, when you do a whole exome, we usually get only 35% of the data in terms of this classification. Rest of the 65% will come to unclassified. So all the clinicians will ask only one thing: what does this mean to me? I'm not convinced because something is sitting there in my patient's case. This is a challenge for bioinformatics in the last 15 years. We used to say that currently this is the knowledge base. This will change in the next 5 years, They were not really convinced because 65% is too high. Now in the last 2 years, this has come to 65%. We know the answer. 35% is unclassified. Now comes the era of AI. So again, as I said, if you go to the deep learning, there are two things again. You have forget the algorithm here. At least from a problem solving thing, from an AI perspective, if you put all the data that is currently available into the deep learning model and build a model and then tweak the model, then the expectation is that you will hit 90% coverage. This is the beauty of deep learning now. So what does this mean is there is also pros and cons here. The number of jobs that will be coming into force will be at that level only, meaning that in the last, this is what we were discussing, IT industry is shifting. Last 8 months, 70,000 IT, the coding people lost their job between Infosys, Wipro and HCL. So the projection in the next 3 years, AI is going to change a lot of the coding jobs. Already a lot of models are coming. LLM is, LLM, the large language model, is changing the whole scientific field itself. So for a youngster who is 21 years now, I think two things I would urge you to do is, if you are looking at a market perspective, then first is do some strong foundation on your either computation biology or data or bioinformatics. One thing is, as a student, you have to understand that every field takes time to master some skill set. At least you have to spend 2 years in a particular skill set to acquire the required skill set. So that pace and that your timing, you have to judge, you have to plan. Nothing will come overnight, but I think one of your faculty told that IT is like hitting a gold and biotech is hitting a diamond, because diamond definitely will outshine, but it takes time to polish and it takes time to go to that valuation. So get first from a biotech, move to, so there are 3 careers now. One is you can continue your biotech into a typical life science career, do a PhD in, there are a lot of new emerging fields like stem cell or whatever from a hardcore biology perspective, or you can move to the flavor of bioinformatics / computational biology. But if you move to bioinformatics, computational biology, then I would suggest you that after 18 months, try to move to AA side and go to read, then understand about the AA and the current thing. So if you look at the current AA in the genome space, let's talk about Google, which everybody knows, right? So any idea what Google has given in the open market in terms of genomics? Any tool name? What is Google doing in Google Genomics? Sorry? DeepMind is the company. Yeah, so DeepMind is a London-based UK company. They had a tie-up with Google. I mean, it's funded by Google. So they started this, they gave this product called AlphaFold, right? So I don't know how many people knows AlphaFold. If you're not, please go and read about AlphaFold. They say that this is completely shaking the whole pharma industry now. Please go and read what is AlphaFold has done to the pharma space and why it is rocking the whole pharma industry. And there is a famous blog by Samir Velankar at European Bioinformatics Institute. He's the team lead at AI division of pharma. Samir is from Maharashtra, but he's a senior scientist there. Samir has given a beautiful article about the role of AI in the era of modern science and modern pharma industry for biotech people. Please go and read about Samir's thing about how DeepMind, EBI and Google work together to give a product like AlphaFold, and why people like Janet Thornton are jumping on it and what it is changing to the market. Number one. Number two is the other product that Google has given and that is shaking the market is Deep Variant. So Deep Variant, again, as I said, how do you classify your unknown mutation? Deep Variant has already given a deep learning model. So they have built a deep learning model on using all the petabytes of human data, and they claim that this is 95% accuracy. F1 is much better. The other major development that is happening is, I am sure NVIDIA, everybody knows. In the world of AI, NVIDIA is, without NVIDIA, nothing can be done because they produce the GPU. And without GPU, there is no AI. So NVIDIA has now tied up with Broad Institute at Boston. So I'm sure people know Broad Institute. If you don't know Broad Institute, please spend 2 days and read about what is Broad Institute doing in the current biotech space / genomic space. And I think Broad Institute is doing a phenomenally superlative work in terms of human healthcare. And NVIDIA has joined with Broad and they have come out with a deep learning algorithm currently that they claim is the best for denoising your base calling qualities. This is at the QC level. So the thing is, if you look at major players like Google, NVIDIA, and there is some announcement from Microsoft, they are already into genomics. So they are definitely into the genomics because they know the market better than anybody. And it's time for you to read about AlphaFold, Deep Variant, DeepMind, Google Genomics, and some of the deployment that AWS has specifically done for genome from Amazon. And currently what I heard is 25% of the revenue for Amazon AWS is coming from life sciences today. That's a huge jump. So revenue wise, people in the industry now see a lot of value from the life science / genome / pharma. So again, please read a lot on AlphaFold. That will give you a perspective about what is the future holding for pharma industry, because pharma in the biotech space is where a lot of money is spent, right? So you have to understand where people are throwing money, what is the next 10 to 20 years holding for us, and what is the skill set required. And as I said, it's not overnight, it takes time. Everybody needs to go skill by skill by skill. So put your foundation very strong, spend a lot of time on the domain, don't lose your domain. Always stay in the domain, but in the problem solving tech space, because that's where the growth is. And AI is definitely it has already penetrated. Today we are looking at LLM model even. So what we are doing currently in Coimbatore is, so we are now moving towards AI. So we are building some models using ChatGPT 4.0. So our idea is that based on our domain and our 10 years data, we want to prove that our model will be slightly better than ChatGPT 4.0, because that's the first benchmark for us. I was really hesitant, and when ChatGPT came, somebody called me from Bangalore and said that, have you looked at ChatGPT? This is going to change. I said, no way, they are not going to touch genome bioinfo. This is a very heavy domain, blah, blah, blah. So two months I was hesitant to look at ChatGPT. Then I think by February I started to look at after noise. It's completely changed. So it has changed my perspective. So I'm now started to learn deep learning and machine learning from scratch. So in June I'm taking a 3 months sabbatical break. I'm going to again learn 3 months full time into AI, because I can't now survive without AI in data side of bioinformatics. So at 57, after 25 years, I'm still learning, and that's the tech space. Until you want to be in the game, you have to adapt to the changes, and the changes will be very fast. If you look at the AI space, right, one day I think 1,000 models are coming. Everybody will publish something, and the Chinese are crazy. So how do you now go into and then survive the market? That's where I think you need to spend time with the industry connect, talk to key opinion leaders, always connect to the industry, and then make sure that you are aligning to what you are trying to do is correct. Otherwise, realign yourself. Don't take your calls on the individual side, because field is changing fast. So I go to my KOL always, even today, and understand that, okay, what is happening today, what should happen in the next 2 years. So realign yourself, learn a lot of skill set. There is a lot of things happening. Only problem is it's like a tsunami. The changes are so fast, they're unbelievable. It's very hard to even digest it. So some of the companies in Bangalore, like Elucidata, their model is very different. They're coming on a very big scale from US and operation at Bangalore. So if you read and then understand how the market is emerging and moving, I think there is plenty of scope, but you should have a lot of patience and put your time and efforts and build your career phase by phase. Thank you all.